Welcome back, everyone. This is Module 5.1. I'm going to be talking a little bit about network modeling in general, and then more specifically, how to do network modeling using the Antimony model specification language in CompuCell. And some people took Herbert Sauer's course last week, and so this will be a review for them. For others, this may be a lightning overview. And I realized from the beginning that there are going to be a lot of things perhaps that are not discussed in detail. And I would encourage you to look at the references that I'm going to refer to. As always, I need to remind you this is live streamed and recorded and available online afterwards. And I'd also like to thank the NIH and NSF funding sources for their support. I should also say that nothing today is going to require, well, nothing in this set of lectures, the first two sets of lectures or the last one, are going to require the diffusion solvers. So even if you're having some issues with the diffusion solvers on your system, you should be able to complete the exercises today. There's a lot to cover in a short amount of time. So if you are not able to complete everything in the confines of this lecture, I recommend reviewing the material, the slides, and the exercises online. And also, last year, the format of the course allowed a slightly longer lecture format. And so if there's some things you'd like more details on, I'd send you to the 2023 videos for the corresponding lectures as well. We're going to be talking about a little bit just generically about the kinds of biological networks that you might want to model. We'll talk a little bit, very little, about the mathematics and some of the questions you might ask when you're thinking about those kind of networks. We're certainly not going to cover everything there. We're going to talk mainly about signaling and gene regulatory networks, although we'll talk a little bit about how you model chemical reactions and metabolic networks. We have used a lot of physiologically based pharmacokinetic models in CompuCell over the years. We're not really going to show you how to build those specifically, but the mathematics is the same. And so the tools that you'll see here are going to be the same that you'd use, even if we don't discuss specific examples of that today. Then we'll introduce Antimony syntax. It's a very simple language for describing networks. We're going to have a program to run Antimony models inside it. There's a typo here. It still has nanoHUB in it. Shouldn't say nanoHUB. We're going to run and load a simple Antimony model. We'll go over the basic syntax of Antimony, which is really so simple you don't need much to learn. And then we're going to do a couple of simple exercises. It's really critical that you download and print that Tellurium Antimony cheat sheet that was provided. If you don't have that printed out and you don't know Antimony, this is going to be difficult because you'll be switching back and forth between things on your screen. And if you can't complete the exercises, I would say it's better to just listen to the lecture and then do the exercises afterwards. Also, in the student materials folder, there is going to be a demo. In this lecture, everything will depend on having that demo loaded. So please download that demo. It'll be called Tellurium V2. Please download that demo to your computer now from the Google Drive. You'll need it for everything we're going to do in this lecture. If you want to learn more, online Tellurium Read the Docs is a great introduction to modeling in Antimony Tellurium. And I realize the Antimony and Tellurium name is a little awkward. Antimony is the model specification language developed by Herbert Sauer, University of Washington. Tellurium is the code that implements that language, which is why there's a bit of an ambiguity. Again, there is a quick guide to Tellurium and libRoadrunner, which is the backend software. And it will say examples of Antimony models. And all the syntax you'll need for building models is there in that one page. that, excuse me, that read the docs here has executable simulations and guides to writing more elaborate antimony models. Those more fancy things than we'll need here today, but it's a great resource, online resource. And then Herbert also wrote a beautiful introductory book, Introduction to Pathway Modeling, that I strongly recommend as well. If you want more information about how to do network modeling, there are many good textbooks, or Yoland's textbook, for example. There are many textbooks and online resources that you can find. Today, we have very limited time, and so we're only going to touch on a few very, very basic ideas. So when we come to biological networks, there are many different kinds of biological networks that we might want to work with. The most familiar might be chemical and metabolic networks that create and destroy chemical components of cells and tissues. Those typically have quite fast timescales on the order of milliseconds to seconds. Those are often modeled as steady state using what are called flux balance equations, which we're not going to talk about today. We have signaling networks that transfer information from outside cells to inside cells and within cells. Those typically operate on time scales of seconds to minutes, and signaling networks are something that people model a lot dynamically. There are gene regulatory networks that switch the expression of genes in the cell, and those operate on time scales of minutes to hours. There's a tendency to draw them as sort of logic diagrams. In fact, they're more complicated than that, but the approximations aren't too bad. And then PBPK models describe the flow of blood and the transport of material in the body if you're going to look at things like toxicants or drug dosing in the body. There are other kinds of network models that one might want to think about. There are, for example, population dynamic models, which are non-spatial models of cells or of animal populations. They may seem very different from these other network models, but mathematically they have the same form, and they can be implemented in the same way we're doing it here. There are also what are called outcome networks or adverse outcome pathways, which are more abstract models that represent transitions between states. For example, you might have a concept of the cell being proliferative and a concept of cell cycle time being regulated, or the health state of a cell, and so on. And those are more phenomenological, perhaps. All of these things, to some extent, are phenomenology. But those are also often represented by rate equations and networks. Fundamentally, the issue here is that all of these things are can be represented by rate equations. In the cell, and we'll come back to this, there'll be another slide, these kinds of networks interact with each other. All three metabolic, signaling, and gene regulatory networks are operating inside cells all the time. But just the way when we're driving a car, we don't really look inside the engine to see how the spark plugs work. Unless we're really interested in diseases involving metabolism, we typically assume that the metabolism, the engine of the cell, is working. Although we might be interested, for example, in limitations of energy production of the cell. And focus on the control, which is the signaling and the gene regulatory networks. The gene regulatory networks essentially control the identity of the cell. And we can ask, how does the identity change when the state of those networks changes? I should say that there are also a lot of other ways of epigenetic modifications in the cell that control how the cell behaves. And those are not really very well understood or very well modeled. And there's a lot of science about cell state and things like cell differentiation that really are not well understood. Surprising how much basic biology is missing today. Signaling networks really talk about how, given some set of gene expression, the states of the cell change. In particular, how does the activity of particular networks change? And metabolic networks essentially control, again, rates of production of components, energy, and other components, and how do those change? We're always thinking about dynamics. So when we're asking questions, typically we're going to ask, how do these network outputs change for particular changes of inputs? We'll come back now to the very first concept, which is a chemical reaction. A chemical reaction is usually written in the form of an arrow diagram. On the left, we have two chemical components, A and B. They react, that's the arrow, to form C and two molecules of D. And so this is called the rate law. The rate of the reaction will depend on the concentration of A, the concentration of B, the temperature, the pH, the pressure, potentially ionic concentrations, many other things. But fundamentally, we have a concept of a chemical reaction. And that concept of a chemical reaction is going to be the way we're going to analyze networks. All networks can be analyzed in this form, even if they're not actually chemical reactions. And we'll see that in a minute. When we write chemical reactions, we have nodes that represent molecular species. We have a choice about whether we represent the state of the node as the amount of the species or the concentration. Typically for chemicals, we use concentrations for chemical reactions because the rates depend on reaction depend on concentration. And so we'll have a concept of multiple reactants coming together in an arrow and producing some set of products. The chemical reactions, by definition, are reversible at some level, but we're going to analyze reactions into forward and backward reactions separately. So we're going to treat reversible chemical reactions as two irreversible chemical reactions because they have different rates. The links, or the arrows, represent the direction and type of the reaction. And we're typically going to write something called the velocity of the reaction, the rate of the reaction, on the arrow. A lot of people are pretty sloppy about this, but whenever you have an arrow, it should be decorated with something that says what that rate law is. And unfortunately, in the literature, you're going to find a lot of inconsistency in the way these things are written down, and we have to live with that. Chemical reactions, of course, conserve mass, but we're often going to assume that we only care about certain components, and so the networks we're going to write down often don't conserve mass. A single forward arrow indicates an irreversible reaction. A double arrow indicates a reversible reaction. VF would be the forward rate of reaction. VR would be the backward rate of reaction. The net rate of reaction would be V forward minus V backward. If there's a positive net rate, we're converting here to ADP into ATP plus AMP. If there were a net negative rate, then we'd be converting ATP plus AMP back to 2-ADP. We're not going to talk a lot about stoichiometry. Stoichiometry simply describes how many molecules are used up in a reaction. Here we have 4 molecules of A plus 1 of B, making 2 of A, 3 of B, and 1 of C. It's allowed to have the same molecule appear on both sides of a reaction. And that happens pretty often in biological reactions. If we want to go further, we have to have some mathematical description of what that rate is. We can translate this chemical reaction here, A plus 2B goes to 3C at a rate VF, which depends on the concentration of A and B, into ordinary differential equations that describe the chemical reaction rates. And these are called rate equations, otherwise called reaction kinetic equations. This one arrow diagram produces 3 first-order ordinary differential equations, one for the rate of change of A, one for the rate of change of B, and one for the rate of change of C. The concept of a rate law is general. The fact that we're writing these things as ordinary differential equations is an approximation which assumes that the concentration of reactants is intermediate, not too high and not too low. If it's too low, we have to think about individual molecules reacting, and we will use stochastic solvers. And sometimes we will ignore the individual levels of chemicals and use what are called Boolean approximations, where we have a probability of changing state. And that's something TJ will talk about later today when he talks about MABUS. Those are used particularly for gene regulatory networks, which typically are on or off for particular states. But they're also sometimes used for signaling networks. If we have some complex reaction, and this is just something made up, it's not a real chemical reaction, we need to have some way of writing the rate of reaction. In general, chemical reactions have relatively complicated rate, but if we assume that the reaction happens with no intermediates, and it's a little bit hard to imagine how that could happen for something this complicated, we often assume, to begin with, something called mass action kinetics. And the derivation of mass action kinetics and how it works is something that you'll have to look up. There are slides in my more detailed materials about this. It's discussed in any introductory chemistry textbook or in Wikipedia. But the assumption is that the rate of reaction is proportional to the concentration of each species raised to the power of the stoichiometric coefficient, and you multiply them all together. So if you have 9 molecules of A, that gives you A to the 9th power. It means it's very sensitively dependent on the concentration of A. If you have 16 molecules of B, it's B to the 16th. And basically, this is derived mathematically from asking the question, if you want all of these molecules, which are moving randomly in space, to be in the same small volume so that they can react, what is the probability that you have 2 molecules of A or 9 molecules of A in the same place? That's proportional to the concentration raised to the 9th power. Actually, mass action kinetics works pretty well. There are some limitations, especially at high concentrations. At low concentrations, it works pretty well. But you have a rate constant here, K, the rate of reaction. That's usually not known for chemical reactions in biology. And those rate constants are hard to measure experimentally. And that's the big problem with writing rate equations. You wind up with a lot of those constants, and the values aren't known. And inferring them in your simulations is not so easy. If I have a chemical reaction, A plus 2B goes to 3C, that gives me immediately a mass action law that the rate of reaction is Kf. I have 1 molecule of A, so I just get a factor of A. 2B gives me B squared, and I can immediately write down then 3 ordinary differential equations for the mass action rates given this arrow diagram. And antimony is basically going to help us do that. We're going to write A plus 2B, arrow 3C, semicolon, then the rate. That's all we're going to do. Chemistry involves not only chemical reactions, and in biology in particular, we have concept of enzymatic interactions and catalysis. If the rate of conversion, say from S1 to S2, depends on a chemical species A is not used up here. A is acting as a catalyst. And so now our rate law will have an A in it. It's always wrong to write that interaction with catalysis using a pointed arrow. And you'll see people do that all the time. It's not right. Our pointed arrows always mean mass transport or mass conversion. So if you see something where there's an arrow pointing to another arrow with a pointed end on it, somebody made a mistake. But I can't help the literature, so you have to be aware of that. So that's the very quick introduction to the mathematics of chemical reactions. Metabolic networks have their own sets of notation. You'll find that there are lots of so-called standards for writing network diagrams. I would say that some of these are pretty widely adopted, some are less adopted. For chemical reactions and metabolic notations, there's something called Systems Biology Graphical Notation, SBGN, that is pretty widely used. It's pretty flexible. There are an awful lot of different kinds of reactions you might want to write. One thing that we have, if we have, for example, a reversible reaction, A goes to B, B goes back to A, is a concept of chemical equilibrium. If we don't have things coming into the system or going out, whatever our initial concentrations of A and B are, over time will reach a steady state. In biology, the only real equilibria are when you're dead. And so we're not usually going to be in chemical equilibrium, but it's an important concept. And in particular, the ratio of the forward and backward rate constants gives us the ratio of concentrations at equilibrium. That's called the equilibrium constant, and it's what normally is measured in experiment. Unfortunately, it doesn't tell us Kf and Kr separately. It only gives us the ratio. In a steady state, we also don't have a time change of A and B, but in this case, we have a continuous creation of A, or input of A, and a continuous destruction of B. And this also will lead to a non-time changing concentration of A and B, but you'll notice there's a continual flux of material into the system. An arrow with a blank on the end, a dangling arrow, represents either the input of material into the system or the destruction of material into something we don't care about. In the real world, there's always mass conservation. A is coming from somewhere, B is going to something. But these dangling arrows mean that we don't, in our models, care about what those are. Okay. That's all I'm going to say about chemical reactions and metabolism. There's a lot to learn, and there are whole books on the subject. But for the purposes of writing down simple reactions, that's all you need to know. When we come to protein signaling networks, these are how cells respond to the outside world, and they also are how signals are sent within the cell. Typically, we will see a molecular receptor on the surface of the cell. That molecular receptor will react inside the cell in a whole variety of ways and send a set of signals that will then do something to the cell. A classic kind of molecular signaling is something called a kinase. The phosphates are the energy currency of the cell, ATP. Adenosine triphosphate is the main energy currency of the cell, although NADP is another one that is used a lot, also has a phosphorus. But a lot of signaling and state transitions in cells are determined by whether phosphate groups are attached to particular proteins or not. And usually a phosphorylated protein is indicated with a star on it. And a kinase is a molecule that attaches a phosphate group to another molecule. A phosphatase is a molecule that takes a phosphate group off of another molecule. And this is a classic signal transduction cascade. One might wonder why you have these complicated cascades. It has to do with the time response and the specificity of reactions, of signaling reactions. Again, I refer you to Herbert's textbook on the subject. But this is a classic signaling cascade. You have a kinase that phosphorylates MAP kinase to phosphorylated MAP kinase. MAP kinase, when phosphorylated, transfers its phosphate to MAP kinase, phosphorylating it. Phosphorylated MAP kinase transfers its phosphate to MAP kinase, and phosphorylated MAP kinase then interacts with a variety of other molecular species, in particular, in this case, regulating cell proliferation. when you're going to look at protein signaling networks, there are a set of typical questions that you want to ask. For example, is the level of a growth factor in the cell's environment or some other chemical in the environment mediated by a surface receptor? Sometimes these growth factors and other modulators go into the cell directly. Estrogen, for example, is transmitted into the cell not through a surface receptor. Does that lead to the cell proliferating or dying? What does it do to the rate of cell growth or death? If we change the number of receptors on the cell surface of the cell, does that increase or decrease the cell's response? For example, does the cell migrate faster or slower? Does a mutation that affects some component of one of these networks mean that the input signal leads to a stronger or a weaker output? There are some mutations that turn these networks on constitutively all the time, independent of the inputs. That's pretty common in cancer. There are other ones that turn off the response, that disable the response. And of course, we might ask, if we perturb one of these networks, does it lead to a change in cell behavior that can then lead to some kind of disease phenotype in the tissue? Signaling networks typically have the form that we just talked about for catalysis. You'll have some molecule S1 that's being converted into S2, and you'll have a signaling molecule A that regulates that. Typically, the rate of reaction of S1 to S2 is an increasing function of A. And that really is just a chemical reaction. S1 goes to S2, or often we don't write S1. This is a shorthand notation. You'll see something that says A lollipop arrow S2. A upregulates or activates S2, and that really is a chemical reaction. Something we don't care about becomes S2, regulated by A, and that's a chemical reaction. Nothing, a dangling arrow goes to S2 at a rate V1 depending on A. You can also have inhibition, which is represented by a T arrow, in which case the more of the inhibitor we have, the less fast the reaction occurs. Again, this can be thought of as nothing goes to whatever our output is with a rate that decreases as the amount of R increases. Again, the critical thing is that A is not and R are not used up in these regulatory interactions. Notice that the arrow from A points to an arrow, whereas the transformation arrow from S1 to S2 points between two chemical species. Typically, we embody our ignorance about the rates of regulation in the form of sigmoids, S-shaped functions. And typically we use Hill functions. Hill functions are a very simple form of sigmoid. We say that the rate of reaction or the rate of production here of species P is the maximum rate of production times F, where F is a function between 0 and 1, depending on the regulator S raised to the nth power divided by k to the n plus s to the n. If S is 0, this is 0. If S gets to be very large, we have s to the n over, this doesn't matter anymore, k to the n doesn't matter, s to the n over s to the n, which is just 1. So we get V max. So this is a function that is 0 when S, our activator, is 0, and it's 1, giving us V max when our activator concentration is very large. The value of K, which is called the Michaelis or the Hill constant, is the concentration at which this is 1/2, and it's called the half maximal concentration. The Hill function has two parameters, K, the half maximal concentration, and N, the exponent. N determines how steep the transition is between being turned off at 0 and being turned on at infinity. If N is 1, you have a different shape. You have what's called a Michaelis shape. If N is bigger than 1, you have a sigmoid, where the value is 0 at 0 and infinite at at 1 at infinity. And as n gets bigger, you get more step-like transitions. There's a corresponding decreasing Hill function, which has simply the form k to the n over k to the n plus s to the n. Again, if s is 0, this is 1. If s is very large, it's 0. And it's a half when s equals k. So if you haven't seen these before, it's okay. You can review them in the literature. This is standard basic chemistry, biochemistry. Okay. It's useful to remember how to deal with these things. You'll see in the biological literature, A promotes S, and you'll interpret that as nothing goes to S with a rate. There's often, in this case, what's called a basal rate, a leakage rate, B1, plus B max A to the n over K to the n plus A to the n. Increasing Hill function, just like that. When you're going to do this in real life, it's very often useful to set all your Ks to 1/2, in which case your chemical concentrations range from 0 up to typical range of 1. They can go above 1, but 0 to 1 basically does the switch. Inhibition has exactly the same form. If you see an inhibition, R inhibits S, you have to write that as V max Kn over Kn plus R to the n. Signaling networks have a number of basic motifs. There are a lot more than this. One is two molecules coming together to form a complex, not forming an irreversible chemical reaction. Another is phosphorylation by kinase, which I've described. Another is the removal of a phosphate by a phosphatase, which I also described. There also are molecular species that promote, like ubiquitination, that promote active degradation of molecular species, sometimes due to ubiquitination, other times due to release of a protecting molecule, other times due to phosphorylation or dephosphorylation. Many proteins have multiple phosphorylation sites. It's not always clear why those multiple sites are there or what the difference is when you bind phosphates to particular sites. When we're talking about gene regulation, we have to come back to the central dogma of biology. That is that we start with DNA. DNA is transcribed into RNA. RNA has some modifications afterwards, but then is translated into protein. We can look at all the steps. We rarely model all of those steps. We typically just do DNA to protein directly. It's a big oversimplification, but it works pretty well much of the time. In fact, you have DNA transcription to RNA with RNA polymerases. RNA to messenger RNA editing out of introns, adding a poly-A tail to the RNA, RNA transport to the ribosome, ribosome being translated into a raw protein, and then protein editing and folding, and then post-translational modification, and then transport of the finished molecule to the final site. And so there actually are a lot of steps between writing a gene as a series of DNA bases and a protein. And people often neglect those intermediate steps. Something that is important to remember is that transcription and translation take time, depending on the length of the gene. And so typically there's a delay of 5 to 20 minutes, depending on the size of the gene and the protein, between the time you start turning on a gene and the time you get any protein at all. And those delays typically are not represented in detail in simulations. Sometimes you can get away with it, sometimes you can't. There are many, many, many molecular species that are involved in transcription and translation. transcription in particular depends on the binding of transcription factors. It also depends on the state of the DNA, whether the DNA is wrapped around histones or not, whether those histones themselves have methyl groups or acetyl groups attached to them, whether those histones are organized in particular ways, where the gene is spatially in the nucleus. All of those things, histone acetylation, deacetylation, methylation, and the architecture of the DNA within the nucleus are very, very poorly understood. And typically, people will neglect those factors in building models of gene regulation. Almost certainly, those are the key things. And so actually we're neglecting, when we talk about systems biology, we don't understand the main things that happen in systems biology. The biology really is not well understood. We're going to typically assume, also in translation, when we're going to translate messenger RNA into protein, we need to have the transfer RNAs and their cargoes present. We're typically going to assume that all of these components that we need for these machines to work are there. That is, we're going to assume our car has enough gasoline and oil in it. But in many diseases and in some even healthy states, you're going to have limitations because of the components not being present. If your car runs out of gas, it doesn't run, even if the engine is well designed. And so we have to think about that sometimes, just to be aware of that. Gene regulatory networks describe how genes are turned on and off, and they typically are drawn like this, trying to make them look like circuit diagrams. In fact, this kind of diagram neglects all of that epigenetic regulation that is so important. It neglects the histone wrap, it neglects acetylation and deacetylation, methylation of the histones. It neglects methylation of the DNA. This is a huge oversimplification. However, it's already pretty complicated, and it often works pretty well. But gene regulatory networks fundamentally change the structure of the signaling and metabolic networks in the cell. So they're sort of master regulators, and they control long-term behaviors like differentiation of stem cells into neurons and so on. It winds up that GRNs by themselves cannot produce stable differentiation. And so there's a bit of a gap because we don't really know how the turning on and turning off of genes with transcription factors, for example, leads to long-term stable changes in DNA architecture through methylation or histone modification. Because genes are often, although not always, either on or off, it's not uncommon to use Boolean on-off models for gene signaling. Kinds of questions you might ask is, how does the rate of production of particular molecular species differ in different cell types or between species? What happens if we mutate a particular gene or knock it in or knock it out, either the gene or its regulatory sequence? How and what are the stable cell types that occur for these kinds of networks? Those are often done using Boolean network analysis. Sometimes it works, sometimes it misses key factors. How does a drug change whether genes are turned on or off? That's critical, for example, in cancer therapies. And how does a molecular perturbant affect behaviors of the cells through this kind of network, whether we're trying to develop a drug or whether we're trying to predict toxicity? Notation, we have this idea of a gene drawn as a little line. An inducer turns the gene production on. It produces messenger RNA. That messenger RNA then is translated into protein. We very often will neglect this messenger RNA step, and we'll simply say that the inducer causes the production of the protein. Remember, there are a lot of intermediate steps, and it takes time for this to happen. And we often neglect that delay in our simulations. Whether we can get away with that or not depends on the particular situation. There are a lot of motifs in gene regulation. You could turn the gene on, and I just said you shouldn't use a pointed arrow to indicate activation. And here's a pointed arrow. This is copied from a textbook. It should have a circle instead of a pointed arrow there. Inhibition with a T arrow. You can have multiple control. And one big problem you face when you have multiple signals coming in is how they combine with each other. Do they add? Do they multiply? Do they do something complicated? If they're both inhibitory or both activating, it's usually pretty easy to combine them. If you have a combination of activation or inhibition, it's not so easy. Genes often produce products that turn on or off other genes. Here, an activator comes in, turns on the blue gene. That blue gene produces a product that turns off the red gene. Genes can produce products that affect themselves. Here, a gene produces a product that turns itself off. Autoinhibition. That's pretty common. You can have things that modulate the effect of transcription factors. Here, a molecule inhibits the activating effect of another molecule. Phosphorylation often has a big effect, either in terms of activating or inhibiting the function of particular molecular species. Again, if you're coming in from a biological background, this is going to be familiar. If you're not, this kind of lightning overview is just so you get to know some of the terms, and you can go back and look them up later. We're going to be focusing today on how to model these things, not on the background, but I have to give you at least a little bit of the language that we're going to use to describe it. We talked about transport networks, these physiologically based pharmacokinetic models. In this case, again, we're primarily talking about blood flow and its transport of material in the body. Lymphatic flow can also be important, and also potentially flow of liquids, for example, in the eye. In the cornea, you have external flow in the tear. You can have gas exchange in the lung. So there are other kinds of flow as well. But PBPK models fundamentally are transport models. They model how the blood flows in the body, how it transports chemicals. The main place you absorb things in the body is the gut, although there's also absorption through the skin and the lungs. The main place you get rid of things in the body is the liver, which metabolizes things and gets rid of them into the bile. The bile in turn goes into the gut. Some of that material is reabsorbed through the gut. Some of that material is excreted in the feces. The kidneys also, if things are water soluble, will get rid of molecules. And so the main inputs in the body are the lungs and the gut. The main outputs are through the kidney and the liver. And you have what's called ADME, absorption, dosimetry, metabolism, and excretion. And that's particularly important if you want to know how long a drug is going to be active in the body, where it's going to go, or if something is toxic, the same question. Many drugs are useful in particular regions, but are highly toxic in others. Chemotherapies, especially cytotoxic chemotherapies, are the classic example of that. Ideally, we would like a cancer therapy to have a high concentration in the tumor, where it's going to kill the tumor cells, and a low concentration, say, in the liver, where it's going to cause damage because it's very toxic. We might ask the question, would attaching that molecule to some kind of binder, for example, putting it in a liposome or attaching it to a nanoparticle, would that help us, or attaching it to an antibody, would that help us get us to the place we want it to be and reduce it getting to places we don't want it to be? Mathematically, We typically have transfer rate equations, which represent transfer functions. Essentially, these represent flow of blood from one region to another. They can also represent exchange of material with the tissues around the blood. They're also first-order linear equations. And if you're interested, there are beautiful textbooks on this that I would refer you to. The point again is that these can all be represented as rate equations, first-order rate equations with arrow diagrams. So the same mathematics will work for all of these different cases. In reality, biological problems always involve all of these kinds of networks. They're going to include chemical transformations. They're going to include signaling. They often will include changes of gene regulation, and they'll also always include transported material. And so the actual regulation of the state of a cell, for example, whether the cell is going to live, die, or reproduce, is going to depend on a whole bunch of signals coming in from the outside through these through these receptors here. Those receptors are going to send signals through a signaling network. Those signaling networks are then going to turn genes on and off through a gene regulatory network. That gene regulatory network is going to then affect the metabolism of the cell. That's going to determine whether the cell dies, stays there, or proliferates. Okay. That is unfortunately all the time I have to go over the biological background of networks. I'm going to ask you now, and I hope everyone has this working now, to download that tellurium V2 code and have it running in Twit. Does everybody have it? The first thing we're going to do is that very simple chemical reaction, X goes to Y with mass action kinetics. X turns into Y at a rate B times X. Y turns into X at a rate C times Y. I need everybody to get that running. Please be patient. I will walk you through what we're going to do. At the moment, we're not going to worry about antimony syntax. Again, just to remember, that simulation is in tellurium V2 in the student materials folder. You can download it here. Hayden, maybe you could put the link for the download in the chat. Open it in Twit, save it as to a convenient location. Again, remembering how to open, click open, point to the file where you've downloaded it. You'll do the unzip and then double click on tellurium CC3D. We're not going to look at the details of the code here. We're only going to look at the network specification, and I'm going to walk you through how antimony specifies network models. That specification is in lines 5 through 15. There's also a line 18 that is going to tell us what we are going to display after we've run the code. So please, everyone, run that code. To do that, remember, you can click on the name of the file, open in Player, and you may have to do a little bit of work with Player to get it to layout, but you should see a plot that looks like this. Perfect. You can neglect the cell window because there are no cells in this simulation. You'll get a blank window for the cells. Good. People are seeing it. Good. Okay. Depending on your computer, these dots may be a little bit too small to see well. The time scale may be too fast, depending on how fast your computer runs. But basically, you're seeing a chemical reaction. X converts to Y, Y converts to X, and you reach an equilibrium. Okay. In line 25, you will see a line. We're not going to talk about what this line does at the moment, except that you will see step size equals, and here it should be set to 1. You can change the interconversion of CompuCell time units to network time units by changing that number. In line 36, you will see a plot, and you'll see a size here for the size of the little icons that the symbols that you're printing. If on your screen those dots are too small, you can make that size bigger. Okay. When you have plots, they're assumed convenience functions. One of them that you might want is if you right-click over the plot and you hit export, some of these convenience functions will actually crash players, so don't try all of them. But if you click on export, you can click export and you can save the image file, or you could save the data set itself. If that crashes your computer, don't use it. I'm not going to promise it'll work on every computer. We're not going to need to save those things now, but I want you to know that at least it should work for you. Okay, I want to spend the rest of our time, and we may overrun by a few minutes on this lecture, on antimony syntax. How do you specify network models? Again, this will be a lightning overview. You'll have that cheat sheet, which will have all of this information written out on it. There really are only 5 things you have to know to write a model in antimony, which is why I like it. The model itself is written as a string, just a Python string, text. If you have carriage returns in Python, you have to use triple quotes. And you're allowed to use either triple single quotes or triple double quotes. But the key thing is that you have to have the same thing at the beginning and the end. So if you have triple single quotes at the beginning, triple double single quotes at the end, triple double quotes at the beginning, triple double quotes at the end. Be careful if you cut and paste from Word or a text editor into your code, because Microsoft Word turns single quotes and double quotes into special characters, which are not single quotes and double quotes. It makes them the little forward and backward quotes to make it look prettier on the page. And the programming languages don't recognize that. So if you cut and paste, you have to redo the quotation marks in the model editor. Avishek?
Yeah, so in the last point, what does it mean by use of carriage returns in the string?
If you have multiple lines within a string, you have to use triple quotes in Python.
Okay.
Okay, good question. Now, the model itself is simply the text inside of that string. The fact that the model is written as a string has some advantages and some disadvantages. It makes debugging a little bit harder. And so if you have typos that cause a crash, you don't always get informative errors. Here's what you need to know for antimony. Double slash or hash specify comments. Those can go at the end of a line or on their own lines. Semicolon specifies breaks between sections. You can tell that this language was based on C because it uses basically C-like syntax. Text followed by colon names things. If you have values colon, that specifies the name of a rate law or an event, and it also creates a variable which allows you to read the state of that variable or event. Okay. If you declare 2 laws with the same name by accident, you don't have to declare names for anything. But if you declare 2 names by accident which are the same, it will overwrite and only the second one will be read. So it's an easy thing to make a mistake when you're naming things. If you accidentally name 2 things the same, the first one will be neglected. Okay, the fundamental concept in antimony is a reaction. It's based on the idea of a chemical reaction. Something goes to something else at a rate. Nothing could be simpler than what you do. If you say X goes to Y at a rate B times X, you type X, maybe name it. Here's reaction J1, colon, name of reaction. X goes to dash caret y, semicolon, b times x, semicolon. So literally, anytime you see an arrow diagram like this, you can instantly type the antimony syntax for it. That's it. And so your rate equation, every rate equation will generate a string. Now, arrow diagrams, as I mentioned, generate multiple ordinary differential equations. And so if I have two chemical reactions, x goes to y at a rate b times x, y goes to x at a rate c times y, that gives me an ordinary differential equation for x. X is destroyed at a rate b times x, created at a rate b times y. Y is created at a rate b times x and destroyed at a rate c times y. You could say, why write arrow diagrams rather than ODEs? The answer is that an arrow diagram can always be unambiguously converted to ODEs. However, the ODEs in general do not allow you to determine what the chemical reactions were that generated them. There are many chemical reactions that would give rise to the same ODEs, but one set of chemical reactions gives rise to a unique set of ODEs. And therefore, you lose information when you go from arrows to ODEs. And as an exercise, I sometimes give a scientifically beautiful paper by John Tyson on cell cycle, where he writes everything as ODEs. And I ask people to say, what were the chemical reactions that gave rise to this? You cannot figure it out. You cannot understand the biology and chemistry of what he was trying to do from the ODEs he presents in the paper. And therefore, the ODEs are less fundamental than the chemical reactions. Again, naming the rate law gives access to the rate. So if I write J1 colon x times y, J1 is equal to b times x. Okay. If I had a complex chemical reaction, 3A plus 5B goes to A plus 2C plus 9D, I just type exactly that. 3A plus 5B goes to A plus 2C plus 9D, semicolon, and then the rate, K A cubed plus B to the fifth. Notice that in antimony, the caret is exponentiation. In Python, it's double star. And so you have to remember that when you exponentiate things, you're going to have two symbols for exponentiation in your code. The simple antimony declaration here gives rise to 4 ODEs. Okay. Any rate constant that you provide, B, C, K, must obligatorily be declared in the antimony definition. If you do not provide an initial value for constants, antimony will complain. It's always a good idea to have a comment that gives the rates, the units of the rates. Is this per minute, per second, so on? Is it moles, millimoles, nanomoles, and so on? Again, double slash or hash are for comments. In our examples, I'm not going to have rates, units defined because these are abstract concepts. But when you're actually modeling things, critical to annotate units. Antimony will default to variables having initial values of 0. That's dangerous. You should always declare the initial values of every variable. You do that simply. B equals 0.1, semicolon, x equals 10, semicolon. Okay. I mentioned before that if we don't care where a molecular species comes from, we can write a dangling arrow. This is a constant rate of creation of A. That would be written E1, semicolon, arrow goes to A, semicolon, K. That gives us the ODE, dA by dt equals K. That means that if we want to write ODEs explicitly, we can always write them as arrow goes to A. That's equivalent to writing dA by dt equals something. If we want to destroy something, we have what's called first order decay kinetics. A goes to nothing at a rate k times A. A goes to nothing at a rate k times A. That generates the rate equation dA by dt equals minus Ka. This will give us the first one, the creation will give us a linear increase of the amount of A. The second will give us an exponential decrease of the amount of A. If you specify the same species in multiple reactions, everything adds up. So here, nothing goes to T at a rate K0. T plus V combine to make I1 at a rate mass action K1 times T times V. I1 decays to D at a rate K2 times I1. Add it all up. dt by dt is equal to K0 minus K1 T times V. dV by dt minus K1 T times V, and so on. It's allowed, although I strongly discourage it, to type ODEs directly using the prime. If you write T prime equals beta times T times V, that will generate the ODE dt by dt equals beta T times V. That's functionally equivalent to writing arrow goes T semicolon beta times T times V. Again, the reason to avoid writing the ODEs directly is that you lose information when you write things as ODEs. And so whenever possible, you should keep the network arrow diagram form. Antimony has a lot of other features. I can only talk about them very briefly. If you're interested, you can look at the cheat sheet, you can look at the online documentation and the 2023 lectures and exercises. We're not going to be able to go over them in detail, but I do want you to know they exist so you know to look them up. Okay. The first thing about antimony is that equal signs are evaluated only when the model is loaded, only once. So if I write A equals 0, and then I write C equals 3 times A, that's going to be equal to 0. C will be equal to 0. If I change the value of A later, the value of C will not change. I can write scaling functions, K2 equals 3 times K1, K3 equals 3 times K2. Those will all be evaluated, but only at the time of load. And that's going to become important because we're going to change values in antimony as we run simulations, and anything with an equal sign will not be updated. Antimony, therefore, we've heard already in Python about, and C, about the difference between a single equals, which is an assignment equals, and a double equals, which is a logical comparison. Antimony has a dynamic functional relationship, colon equals, that says continually check if the right-hand side of this is updated and make the update. That's called the functional relationship. If I write K2 colon equals 3 times K1, and I change the value of K1, now the value of K2 will change. Now, there's some limitations about how you can do that, which we're not going to talk about right now. Another thing that happens is we've talked a little bit about the difference between dynamics, things that change continuously in time, and events, things that happen in specific times. Antimony has a concept of events. Very simple to specify, just like reactions. Event name, E1, colon, at, specifying the event, condition, for example, time greater than 15, colon, what happens? A increases by 15 units. We can have multiple events slaved to the same trigger. We're not going to use events much inside of CompuCell, but they're useful to know about. There's a concept of a trigger. The event occurs whenever the trigger goes from false to true. So if the trigger is time greater than 15, that only happens once. If the trigger is sign of time being bigger than a half, that will oscillate when sign of time goes from being bigger than a half to smaller than a half, nothing happens. When it goes back to being bigger than a half, it'll add 0.5 to B a second time. Okay. That's all you need to know about antimony syntax. Let's write a couple of exercises. The first exercise is go into your code, look at lines 10 and 11, which specify the rates of the forward and backward reactions between X and Y. Okay, change the value of C, which was initially 0.01 to 0.1, and run it, and ask the question, do the steady states of X and Y change? And once you've done that, you could try a couple of different values of B and C. Okay, I want to make sure everybody has this working. Notice that the equilibrium values change. So if you change B and C, the ratio, the equilibria of X and Y depend on the ratio B to C. So if you now make, for example, if you make B and C equal to each other, make C equal to 0.1, you should get the values of X and Y are equal to each other at equilibrium. Everybody get that? Somebody says CC3D is crashing, then you'll probably have a syntax error in your code. If somebody wants to help people who are having trouble in a breakout room, that would be great, or you can wait till the end of the lecture. All right. As you change that ratio of B and C, you'll see X star over Y star, the equilibrium values will change. X equilibrium over Y equilibrium is equal to C over B. As long as you're getting a plot that looks like this, may not have a black background depending on your computer, but as long as you're getting a plot like this where you have a black diagram which has a red line and a blue line on it, it's working. So you do need to play around with laying out your screen a little bit to get things to be about the right size and shape. Depending on your computer screen, the size of the dots is determined in line 36. And so if you change the size to be bigger, you'll get bigger dots. And if the simulation is running too fast, you could make your step size smaller, make it 0.1, for example, in line 25. Again, we'll come back to this code, how to control this code in a minute. But this is controlling the display options, essentially. Okay, I mean, you can always, on a Mac or a Windows machine, you can always pull the boundaries of your windows to resize them. But in Windows, the tile will resize automatically for you. One thing to do is when you start the simulation, instead of hitting run, hit step, and then adjust the windows at the first time step, and then run it. That may help. Okay, let's do one simple exercise here. We are going to take the code that we have now, and we are going to write new code, which is now going to have 3 chemical reactions. You're going to make J1, A goes to B at a rate Ka times A. B goes to C at a rate Kb times B. C goes to D at a rate Kc times C. To do that, you're going to have to change this set of lines, lines 6 and 7. You're going to add one line and change those 2 lines. You're going to have to add definitions of the rate constants, Ka, Kb, and Kc, in lines 10, 11, and you'll have to add a line. You'll have to define the initial values for A, B, C, and D in lines 14 and 15, and you'll make it 10, 0, 0, and 0. And then finally, in line 18, instead of just displaying X and Y, you want to display A, B, C, and D. And you can use other color names, simple color names like green and blue, and green and orange, if you like. If you're having trouble with this, exercise 5_1_1_ sequential reactions is the completed code. So you have to change 1 line, 2 lines, 3 lines, 4 lines, 5 lines, 6, 7, 8, 9, 10, 11 lines. I'll give people a minute. If people get ahead, you can try changing the values of Ka, Kb, and Kc. And it's interesting to make them 1, 10, and 100, and 1, 0.01, 0.1, and 0.01. Okay. I'll give people 5 minutes to try that. Or until, if everybody gets it beforehand, I can do it sooner than. Again, if you feel stuck, please download that completed one and try it out. Yes, sometimes there's some comments about resizing windows. Sometimes you do have to resize windows manually. Again, if you hit the step function to launch the code instead of the run function, then you can do the resizing at the beginning and see what happens. Because you're not running that many steps in this, you may actually, instead of hitting run at all, you may just go step, 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 step through it. The key thing here is to familiarize yourself with how to write these simple rate laws. Are there any questions about this little exercise?
Are any colors not defined in antimony?
I don't know. Probably if you type puce, it won't work.
I type black and green. I'm getting some weird thing.
Well, I don't think black is probably a color you want to use, but green works, orange works, red works, yellow works, pink works. I haven't explored all of the possible colors. The basic colors work. People are getting results here. Again, if you have those basic results working, I encourage you to try these little extensions where you change the values of Ka, B, K, and C. You could also try changing the initial values of B and C and D and see what happens. In this chemical reaction, if you look at it carefully, you'll notice that no matter where you start, you always wind up with no A, no B, and no C, and everything turns into D. There's a question, what are the variables we're changing in line 1? I think you mean step 1, perhaps? You are changing the yellow. That's why it's coded like this. Instead of X goes to Y and Y goes to X, you're going to write A goes to B goes to C, and C goes to D. Step 4, you're going to have to, here you're displaying X and Y. You want to display at least A and B. So you would change X to A, Y to B. And then ideally you would type comma, quote, C, colon, orange, comma, D, colon, green, or something like that. Let me show you code that works. J1, A goes to B, semicolon, Ka times A. J2, B goes to C, semicolon, Kd times B. J3, C goes to D, semicolon, Kc times C. Ka equals 1, Kb equals 1, Kc equals 1. A equals 10, B 0, C 0, D 10, 0. A colon red, B colon blue, C colon green, D colon orange. We're not going to be linking chemical concentrations to fields now, but I'll show you how to do that in principle in the next exercise, and then you'll do it tomorrow. People are getting it. Somebody says you don't see the D plot. If you have in line 22, if you have comma D colon orange, it should display. Anything listed in line 22 will display. But you have to add it to line 22. Okay, there's some antimony annoyances that you're going to run into. There are multiple reserved words which you cannot use as variable or parameter names. Some of those are pretty obvious. like time. If you do, tellurium is going to die, and it won't give informative errors when it dies. On the other hand, gamma is also a reserved word, and that may not be so obvious. So I strongly recommend, if you have a name like delta or gamma, to put a number after the end. Gamma is a reserved word. Gamma 1 is legal. Missing or extra parentheses or improper separators, you're missing the semicolons, tend to make tellurium die with uninformative errors. If you're going to debug antimony, the best thing to do, and it's boring, is to comment out the entire model with hashes at the beginning of every line. Make sure it loads without error. Then uncomment the variable initializations and make sure it loads without error. Then uncomment the parameter initializations and make sure it loads without error. If you're using events, which we haven't really talked about, uncomment them one at a time. And then uncomment the rate laws one at a time starting from the top. It's not very elegant, but that's the fastest way to catch typos in antimony. Almost always, if you have a crash, it's because you have a typo in the antimony, an illegal syntax in the antimony. Okay. Let's take that code we just wrote, add one more line to it. D goes to A at a rate KD times D. You're going to have to add one line after line 8 here for the D equation, and one line after line 13 to give the rate K times D. You don't have to change 22 because you're still displaying A, B, C, and D. You don't have to change the initializations here. Why don't you do that and have somebody tell me what changed? Remember that these things are case sensitive, so you have to be a little careful about that. It's also pretty easy to write a colon instead of a semicolon or have a comma instead of a semicolon, and that will cause errors. You'll notice this one actually reaches a chemical equilibrium. The last one didn't. And so it's very similar, but instead of having everything turned into D, you're going to get, if these rate constants are all equal, you get an equal amount of A, B, C, and D. So here's the comparison. In the new one, you get an equal amount of A, B, C, and D. In the first one, you got everything turning into D. And you can play with this and explore how the rate constants and initial values change the results. We're not going to have time to do that today, unfortunately. There's one other thing I want to talk about, and this is a very big subject that we're only going to talk about very briefly, which is that, again, it's a reason to use arrow diagrams rather than ODEs. Chemical reactions actually occur through the interaction of individual molecules. They're discrete processes, and so writing them as continua with concentrations is an approximation. Now, if we're talking about reactions involving water or ATP, where you have a lot of it in the cell, then a continuum approximation makes sense. But many important biomolecules occur at very, very low concentrations. You have one copy of the DNA or two copies of the DNA per chromosome. You have, for some transcription factors, 5 or 10 copies at most in the cell. If you have very low concentrations of molecules, then the stochasticity of the molecules finding each other and reacting is important. There are many algorithms that try to approximate that. The most famous one is called Gillespie. And in this case, the rate of reaction is the probability per unit time that one molecular reaction occurs in the volume of interest. One thing you have to worry about with Gillespie is that for chemical concentrations, we typically normalize everything to 1. In Gillespie, we're actually counting molecules, and so we have to use numbers bigger than 1 to make things work. The Gillespie solver actually does individual molecular reactions, And so it can be a lot slower than the ODEs. And so if we have large numbers of molecules, you don't want to use it. You also have to rescale the step size in the integrator based on the number of molecules. If we want to use the Gillespie model, we use a line that says add free-floating antimony model. We've already modified this once by changing the step size. We would add comma integrator equals Gillespie. And that will switch us from the continuous ODE solver to the stochastic Gillespie solver. I'm not going to talk about this. You can refer to this about how to do the time rescaling later on. In our last, or a little over time already, but if people feel like doing this, you can change the initial value in your simulation from 10 to 100 here in line 18. And in line 31, add that comma integrator equals Gillespie and run it and see what happens. If you don't get this at the moment, keep working on the previous problem. But for the people who got this working, try it out. If you want to load the code, it's exercise 5_1_4 Gillespie. And I'd like to ask, if you do it, how does it differ? And then you could also try changing the initial value here of A, try 10, try 100, and try 1,000, and see how it differs. You see, it's not, if you make it 1,000, it's not that different from the continuum approximation. If you make it 10, it looks very different. Go ahead.
Yeah. Is this just adding basically like a white noise to the ODE part? Is this what it's doing?
No, it is not the same as that. In the case of having very large molecular concentrations, it is similar to that. But this is actually a discrete model. It actually does a stochastic process where at each time step, it looks at the local concentrations, rolls the dice, and says, did a single reaction occur at each time step?
Perfect.
The Gillespie model is quite beautiful. There are a lot of other stochastic approximations to chemical reactions. Gillespie is only the most widely used one. And there's rather a lot of mathematics and argument, in particular because Gillespie is so slow. There's been a lot of work done, which we don't support here, about trying to develop hybrid methods that combine stochastic and deterministic models. It is possible to add noise to these systems with the ODE systems. I'm not going to be able to show you how to do that today, but
No,
if you're interested,
just with a simple code like this, pretty cool already with the Gillespie. Yeah.
Gillespie is the standard one that people use at least to start with. Omar, question.
Hello. Yeah. Have you tried to implement Gillespie modified like tau leaping or something like that? Because I think that kind of solvers are quicker than pure Gillespie.
No, we only support Gillespie and ODEs.
Okay.
This afternoon, you'll see stochastic Boolean models are available. In principle, if you have a solver that you like, because these solvers are fundamentally Python libraries, if you have a Python library that implements the solver you want, you can load it into CompuCell and use it. So there's nothing preventing you from doing that. But built into CompuCell libRoadrunner, which is the backend for Tellurium and for CompuCell, and also for TissueForge and also for Physicel, the default solvers are ODE and Gillespie. There are a lot of very nice alternative solvers, but those are not those you'd have to pull in yourself. You're free to pull in anything that's a Python library you can pull in and use. So there's no reason you can't, but they're not built-in functions in CompuCell. if we found one that people really liked, we'd probably try to deploy it as part of CompuCell. So if you have suggestions, we can try that. All right, so you should have gotten something that looks like this. As you make the initial number of A smaller, the thing will get noisier. As you make A larger, it'll get smoother and look more like the ODE system plus noise, as Omar was just saying. All right. If you want to extend it, there's a lot you can do here. We've only, in an hour and a half, only been able to touch on a few basic ideas here. The key thing I want you to know, again, is how to write a model in terms of antimony string, the basic idea of expressing chemical reactions in terms of these arrow diagrams. And then the next lecture, we'll talk about how we actually do this integration between the network models and CompuCell. So here was actually the simulation where we changed the initial values. 10, this was probably 10. I can't see this, maybe 100, 1,000, 10,000. Okay. So that's all we're going to do for today. I realize that that's a lightning overview of this. For people who've done network modeling before, it'll be review. For people who haven't done network modeling before, it may be a little fast, but we'll have plenty of opportunities to come back and go over these things again. As I said before, we're learning a lot of languages. And so when you learn a language, you have to hear the same concepts repeatedly to be able to master them. And so we're going to call that here. I'm going to do the module poll, and then we'll take a short break and come back and talk about how to integrate these kinds of simulations with CompuCell. Again, Herbert's workshop is a great way to learn this material. Last year we had a little bit more time, and so there are lectures that go in more detail. I also have a set of online lectures which spend a whole semester's worth of time covering this material. And so I could point you to that lecture set if you're interested. Ramon, please go ahead.
Yeah, thank you very much for the presentation. I had a question, maybe it's a naive question, but is there any way or any way to solve these systems of differential equations, providing the time points where you want the solver to solve the system? Because I have a model or data that go from pico to 1,000 seconds. So how I usually get this data from other Monte Carlo softwares is to get the data in a logarithmic scale, scale of time. So I don't know if it will be possible to solve the systems of differential equations in CompuCell or in Antimony, providing the time point instead of setting just the step size.
So I'm trying to think if it's supported in LibRoadrunner. Fundamentally, the answer is no, because you typically are running these in the context of multicellular systems where the timescales are set by the behaviors of the cells. In principle, you can run inside of CompuCell multiple network models, and you could set different timescales for each of those network models. So you could run a network model on a short time scale and another one on a long time scale. One problem is that I don't believe you can update the network models separately from each other. And so I think that you could set the number of steps per Monte Carlo step separately for multiple models, but nevertheless, per Monte Carlo step, they would all update whatever their particular time setting is. So there isn't the kind of adaptive time setting that you're looking at. It would be a nice thing to have, but it would be pretty complicated to set up. And again, I think the issue here is if you have a library that you like that does that, I would recommend loading it into CompuCell and using that instead of the built-in libraries. Otherwise, what you'd probably do is you'd take the fast timescale one and run it with a flux balance. You'd assume it's equilibrated, and then and then use the outputs of that as the input parameters, the boundary parameters for the slower timescale ones. If you have separation of timescales, you never want to evaluate the fast timescales explicitly during a run. So the next module is basically going to be now focusing. We've done a little bit on antimony syntax. The next module will now be focusing on how to integrate that antimony syntax with ComputerSum.